Hi, my name is Sophia, and today I will be introducing the bacteria Campylobacter J. Campylobacter J, also known as Campylobacter jejuni, is a foodborne pathogen contracted through the consuming of contaminated water or foods, such as unpasteurized milk, fresh produce, and raw poultry. It is a polar flagellated bacteria with a helical cell body. The shape of Campylobacter jejuni allows it to have very high motility in viscous solutions, such as blood, as well as in enhanced colonization ability in comparison to other types of bacteria. Campylobacter jejuni specifically colonizes the mucus layer and intestinal crypts of the small intestine and invades intestinal epithelial cells and monocytes, which are a type of white blood cell. Symptoms of a Campylobacter jejuni infection include nausea, vomiting, diarrhea, fever, and abdominal cramps. Although Campylobacter jejuni invades both monocytes and intestinal epithelial cells, today we will be investigating the invasion of intestinal epithelial cells and how cell signaling is affected. To begin the process of invading the intestinal epithelium, proteins on the Campylobacter jejuni bacteria, circled in blue, called CADF and FLPA adhesions, serve as ligands that will be received by a membrane-bound receptor that is circled in purple, called fibronectin. The receptor must be membrane-bound in this case, instead of internal, because of the bacteria itself being polar and unable to pass through the membrane to access an internal receptor. Once the ligands are bound by the fibronectin receptors, CIAD proteins, which are circled in red, will be released from the inside of the Campylobacter jejuni bacteria into the host cell. Once the CIAD proteins from inside of the bacteria invade the host cell, it will set off a phosphorylation cascade of focal adhesion with the help of the enzyme kinase, and thus reorganize the host cell's actin, which is another protein that provides the cell with structure and support. This can be seen in the photo displayed on the left side of the slide, as CIAD inhibits one protein that inhibits the reorganization of actin, while simultaneously activating a second protein that usually activates the reorganization of actin. The reorganization of actin will weaken the cell structure and therefore allow the bacteria to be engulfed by the host cell during the cellular response. The cellular response is the engulfing of the Campylobacter jejuni bacteria itself, which would then produce its own toxins to disrupt the host cell's cell cycle and then cause it to swell up. The correct signaling mechanism would be everything that I just described in the previous slides. In short, the fibronectin receptor of the host cell would need to bind with the ligands, which are the CADF and FLPA adhesions, which are attached to the Campylobacter jejuni bacteria, and allow the CIAD proteins from inside of the bacteria to be released into the host cell. With the help of the phosphorylation cascade that happens as a result of the binding of the signal and the receptor and the presence of CIAD, actin would then be reorganized within the host cell, and the bacteria would be engulfed and enabled to produce toxins as a cellular response. However, if the mechanism were to be faulty, it could hypothetically be caused by the absence of ligands on the bacteria or faulty receptors on the host cell. If ligands were absent, there would be no effect on the ability of Campylobacter jejuni to adhere to the host cell itself. However, there would be a decrease in the likelihood of the host cell engulfing the bacteria and allowing toxins to be produced afterwards. If the host cell has faulty fibronectin receptors, there wouldn't be successful binding between signals and the receptors, and no transduction or cellular response would occur as a result of that unsuccessful binding. Thank you so much for listening to my talk on Campylobacter jejuni.